The following is a conversation with Dr. Petra Louis, Senior Research Fellow at the Rowett Institute for Nutrition and Health at the University of Aberdeen. I was really excited to have Petra on this podcast. She was actually the first academic working in the field of microbiome science that I spoke to about interbiotics. And I'll never forget that meeting that I had with her, where I shared my vision for transforming the microbiome through full spectrum therapeutics in what was then the old Rowett Institute near the airport in Aberdeen. Because I've known Petra for so long, this was a really engaging conversation that I think you'd expect friends to have, actually. And we entertained various different subjects in a fun but also quite serious manner. And we got into some nitty-gritty fun topics with regards to diets, supplements, fibre, all the things that we've talked about before in interbiotics, but going into quite some significant detail. Since the conversation, I've actually rethought how I manage my supplement stack. So for those of you that are taking supplements and interested in longevity and health span, this would be a good episode for you. For those that are supporting the podcast, I just wanted to shout out and say thank you very much. I've recently reviewed my metrics and was really pleasantly surprised actually to see how many people were listening to each episode, and also how far and wide my listener base was from around the world. Quite stunned actually to have listeners as far away as Singapore and other parts of Asia. So just want to take this opportunity to say thank you to you all for supporting the vision and the mission. I really appreciate it. The best way to support the podcast is to listen, but also to share with your friends, to like and subscribe, and to give a 5-star review if you haven't already on your favourite podcast app. I'm open to suggestions for how to make things better looking forwards into the future, so please reach out to me on socials or directly through the email address james@insidematters.health. This is Inside Matters. My name is Dr. James McElroy. I hope you enjoy it. How did you get interested in the field of the microbiome?
So when I started my research career in Germany, where I did a diploma degree in biology, I ended up in a lab that was working more on microbial biotechnology, and specifically on compounds that are made by microbes that live in high salt environments, so salt lakes such as the Dead Sea, let's say. So these environments are essentially very, very dry environments. Because of the high salt concentration, the cells basically dry out. So very few organisms can actually live in these extremely high salt conditions. And we're talking much higher than seawater, so maybe 10 times more salt in a lake like the Dead Sea. I think it will be more than 10 times. But anyway, it's much, much saltier than seawater. So the organisms that can actually survive and even grow and multiply in these conditions, they need to have mechanisms that they can actually survive there. And one way of doing this is by accumulating in very high concentrations molecules inside, so that there's osmotic balance across the membrane. And one strategy is to use these organic molecules, and they're called compatible solutes because they are, despite the fact that they're present in very high concentrations inside the cell, they're still compatible with the cell being still able to do its cellular functions. So they're not really messing with the biochemistry that happens inside cells to let them grow and divide and all this.
Why is that? Have they got like special compartments within their membrane to sort of keep them separate, or is it just they're at such low concentrations they don't interfere with the normal physiology?
No, the concentration is extremely high. It's really more that they seem to not interfere with enzyme function, for example. So there are actually some bacteria, or there are some microbes, not bacteria, archaea, that actually let salt in. May not be exactly the same salts that are outside, but they may have a lot of potassium inside and chloride. But they had to really adapt their protein machinery completely. So you can see that actually the protein composition is different. They have many more charged amino acids to sort of neutralize these salts that are inside. But they're kind of obligate extremophiles. Extremophiles are organisms that can live in extreme conditions, whereas a lot of the bacteria use these organic compatible solutes, and they're just less inhibitory to enzymes. They work better with our normal enzyme apparatus.
Got it.
And our organic molecules that make up our cells. And I'm saying our, I mean obviously bacteria here.
Got it.
So that's just a different way of dealing with it. And these all sorts of organic molecules. One, for example, is glycine betaine. And that's also a molecule that I think we have in our metabolism somewhere. So they sort of adopted different molecules. Other ones use also glutamate, which is an amino acid.
Yep.
And then in microbes specifically, we worked on a specific group of compounds where the compounds had been found. But in those days, we didn't really know the genetics. We knew the bacteria who made it. So in my PhD, I looked at the molecular biology of finding these genes that make these compounds. But beforehand, in my diploma, I actually worked on a much more biotechnology applied project where I was looking at, can these solutes be used, applied, to preserve organic molecules or organisms. So somebody else was looking at, can they preserve enzymes, for biotech companies? And I was looking at, can they preserve organisms? And you know what the organism was?
I'm going to hedge a guess. E. coli? Yes, it's E. coli.
E. coli, Nissel, 1917.
Oh, wow, no way. Only proper microbiome enthusiasts will understand the significance of what you're talking about. But I'm glad I got it right. I was thinking E. coli because it's like the workhorse of biotechnology, and then we've got E. coli Nissel.
Good thought.
That kind of makes sense because, well, it's in Germany.
Yes.
And they love E. coli Nissel in Germany. So for the listener, sort of try and contextualize all of that. Mm-hmm. Yeah.
So a previous PhD student in the group had gone on to that company that manufactures E. coli in 1917.
Got it.
And Nissel's 1917. And so this is a strain of E. coli which is used as a so-called probiotic. It's a live bacterium that people can consume to keep their guts healthy, and particularly it's often used for traveling. Or also diarrhea. So I have had another of my mates from Germany who later on worked in the company, and he always had some in the fridge at home for his kids. So if the kids were getting an upset stomach, he would have it ready there. And you know the story where it comes from?
No.
It was isolated from a soldier in 1917. Who went to, I can't remember exactly where he went, but actually as part of the war. So I never read this somewhere, but that's the story I still remember from the day.
Why was the soldier selected? Do we know?
Because he never got
ill.
Any illness. Yes.
Oh, I love this story.
So they were
that's actually a next-gen probiotic. They were on it back in 1917 because one of the critiques is, lactobacillus, bifido, the reason that they're in quote-unquote probiotics is because they're stable in yogurt and cheese and milk. But this sounds a bit more sophisticated. Somebody at some point was like, that soldier, he's so healthy.
I know.
And they took a stool sample and they isolated it, and that was then what has become quite a popular probiotic.
Yeah.
Now, one question. Do you take E. coli Nissel 1917?
I don't really know.
You don't take any probiotics? Or
No, I don't need sort of probiotics that are marketed as, I don't go to the pharmacy and buy probiotics. It's very difficult to get non-probiotic yogurts these days. So I do quite like a yogurt once in a while, and if it's probiotics in it, that's fine with me. It doesn't bother me, but I'm not buying it for the probiotics. I'm looking more for, I want sort of natural yogurt. I don't want the zero fat and the zero sugar because it has some other stuff in it that I don't want. So I don't mind the probiotics, but I don't take them because I think as a healthy person with a healthy diet, I don't need a probiotic. But obviously for certain indications, for certain problems that people have, I think probiotics will be one avenue to take that will help some people. Mm-hmm. Absolutely.
Now, back to E. coli 1917 and your journey into the microbiome. So what happened next?
So after this whole, the years in the extremophile field, I wanted to move on to another lab, and I wanted to work on something related but not quite the same. So I actually ended up in the end, after looking at options in Aberdeen, in a lab that was working on E. coli, not Nissel 1917, just E. coli generally, and stress regulation. So, I moved away from that extremophile, which was fun to work with, but we had nothing. And also in those days, we didn't tend to have genome sequences and all this. So we had much less information about the organisms then. We're talking sort of late 90s here. But for E. coli, there was already much more. There were more tools and all this. So, with my organism, I tried for the first 2 years of my PhD to get genetic transfer methods, which you need if you want to really look into the detail of gene regulation, molecular biology. And I didn't succeed. So I had several streams going. My PhD went okay, but that thing didn't work out. So I was quite keen on going into a lab where I can really get more into the nitty-gritty on a sort of model organism.
Yep.
But it wasn't an extremophile in that sense. So I did a postdoc there, moved then on briefly to another postdoc.
Why is E. coli such an interesting organism?
That's a good question. I think part of it is probably serendipity. I don't know. I've never really looked into the details, but it's quite easy to grow. Yeah. It is a gut bacterium, but it also grows very happily outside. It can grow in the presence of oxygen, in the absence of oxygen. It doesn't have too many needs. It grows very fast. Also, obviously, different strains will be different. They're not all the same.
And they can be vastly different, right? You can have
absolutely.
The 1917, and then you have 0157, which can give you, really advanced kidney disease and maybe kill you.
Yeah, absolutely.
So is it kind of unique in that regard with so much variation across the strains, or is it just we've studied it lots? And
it's a good question. Again, I haven't really looked into this in detail. I think there's some species where probably most of the strains are pathogenic, as in disease-causing or not. I'm not aware, for example, of any bifidobacterium that has caused disease.
Likewise.
I don't think so. And I'm now, for example, looking at listeria as another sort of foodborne pathogen.
Yep.
I'm not aware of non-pathogenic listeria, but now that I'm saying this, I'm not so sure. Microbiology is very complex.
Yeah,
so
incredibly complex.
It's probably partially also because it has been studied so extensively, but also with its lifestyle being very versatile, it will have had evolutionary a lot of opportunities to adapt to different niches, because it can just end up in a different environment and is already so versatile that it will not immediately be killed. And then it maybe has time to adapt to these different environments.
Find its way into some other one. Yeah, that makes sense. So, sorry, back to your work.
Yes.
I'm sure we'll talk a lot about E. coli and why it's important. I guess just one more point. Is there still a lot we don't know about E. coli? Is there still a lot we don't know about microorganisms?
Mm-hmm.
Yeah,
I think so. So I left that. I had another brief postdoc, but then I moved on to the gut field in 2002, I think. So it's a long time ago for me to have looked at E. coli in detail. Enough. I can't really tell you, I never look nowadays at the databases in E. coli. But in principle, I think even for bacteria like E. coli, we still don't know, if you look at the genome, for example, we still don't know what all the genes do. And some bacteria will be more understood than others. And I'm sure in a model organism like E. coli, there are groups who either have or are in the process of knocking out, so basically destroying individual genes to see what does this gene do. But I assume that still in E. coli, there will be a lot of genes we can see from the sequence in a genome where a gene should be by looking at basic characteristics of the DNA sequence, basically. But then we know, this should be a gene, but we may not know what it does. And, some bacteria like E. coli, also maybe some yeasts like Saccharomyces cerevisiae, a lot of this very fundamental work is done. But I think there's still, for example, genes out there that we don't know what they're doing.
So there's still some dark matter in the genome of microbes.
I think so.
Just like there is in our own genome, right? There's lots we don't understand in terms of how does it work. And, we're like 98% genetically similar to a banana. But we're very different to a banana, right? So there's a lot that we don't understand. I think maybe we should touch on the genetic diversity element just a little bit, because my understanding is that there's vastly more genetic diversity in the gut microbiome than there is in the human genome, like 1 or 2 orders of magnitude. And when you got started in the field, where were we, if you like, in 2002, I think it's you said.
Yeah.
And if you can also contextualize it in what we were talking about before we started, around the hype. Where were we on the hype cycle at that time,
Yeah. So when I moved on, that position at the Rowett Institute became available, and I was very intrigued by it. So I applied for it. And for me, it was really quite a big change, because up to then I had always worked on like one organism. That was my pet, and I was trying to work out something about how it functions, what it does.
Right.
And this obviously was a huge step change. And also, I'd never worked with anaerobes before, so I went from working with aerobic organisms, which are organisms that live in the presence of oxygen, to strict anaerobes, which is technically quite a big change. So strict anaerobes are those, most of the bacteria in the gut are in the lower intestine at least, are strict anaerobes, which means they are killed by oxygen. So they would not survive on this desk.
For how long?
It's different. So I think my colleague Sylvia Duncan has done some studies. So she plated out different bacteria in an anaerobic cabinet, which is basically a box where you can control the gases that are inside, and you stick your hands in through gloves to manipulate them. And she literally plated them out, took them out, put them back in. And I think the quickest she could do this was a minute, I think. And some of them were literally had already gone down by logs. Yes. And others, you can see the sort of death curve over time, is a bit less dramatic. So it really depends on the bacterium you're looking at. So as a general rule, the Firmicutes, the gram-positive bacteria, tend to be more oxygen sensitive, but within those you find differences, and the Bacteroides maybe cope a bit better with
Do we know how they die? Like, what happens? Do they
Yeah, basically,
explode,
oxygen
sizzle?
is toxic for organisms. It's toxic for us too. Did you not know that?
I better stop doing my Wim Hof hyperventilation then every morning.
Oh yeah, I'm not sure
Every
that's
morning.
good.
I do it every morning. I'm not joking.
Really?
Yeah, I heard it's good. Every morning. I'm not
It
joking.
reminds me of Howard and Maude.
Even in weird places, and people look at me and they go, "What is that man doing? Is he having some sort of anxiety?" And
Are you doing
Yeah, I think it's called Tibetan fire breathing.
Okay.
So you hyperventilate for like 30 or 40 breaths. Then you squeeze. It's like a Valsalva maneuver. And then you hold your breath for as long as possible. And then you repeat. Now, what are the benefits? Well, you're hyper-oxygenating your blood. You're maybe changing the acidity, alkalinity. Then you're holding your breath and it's probably becoming more acidic. There's some element of deep relaxation that comes with this. Probably to do with the vagus nerve, I think.
Okay.
And Wim Hof, who's a sort of Western, modern-day superhuman... Do you know who I'm talking about? The Iceman.
Oh, yeah, I do know.
Iceman, yeah. He is a medical and scientific marvel. So you'll enjoy this one, I think, because you're a microbiologist. So when he started coming out and doing his things, people thought, this is just Wim Hof. He's able to be in an ice bath for like an hour. It should be impossible. He's ran marathons with no additional water. He climbs mountains with just shorts on. He's also able to somehow train his immune system through this breathing in the cold. So he's been injected with endotoxin, and there's been no physiological response. Just super high antibody titers. Okay. And then they said, it's just you, Wim. You're special. He said, no, I'm not. I can teach other people. So there is an experiment which I can send you. I think it was published in Nature, where they split the groups. One had the Wim Hof training, the other didn't have the Wim Hof training. They were given endotoxin. And in Wim's group, who'd have his training, they didn't have a systemic inflammatory response syndrome.
Wow.
Which seems impossible.
Yeah.
But anyways.
So is this controlled through the mind?
Yeah.
Must be.
So breathing is both autonomic and conscious. And I think the deep breathing and control over your breath allows you to start to tap into your autonomic nervous system. It has some impact on vagus tone and vagus control. What it does to the immune system, I don't think we know, but I think it does good stuff. Anecdotally, get sick much less often. Anecdotally improves resting heart rate. I think it's quite good for anxiety and things like that, whether it's the cold or the breathing, I don't actually know. Really recommend it. But you just told me oxygen is toxic, so...
Yes. No, first thing you tell me, how long do you do that for?
Right, so probably for 15 minutes every day.
So you go through cycles?
You go through cycles. So it's like 2 minutes and then hold your breath for 2 minutes. 2 minutes, hold your breath for 2 minutes. Wow. Sometimes 3 minutes. Sometimes 3 minutes. And this is a big movement. Now, I do know that the actual oxygen content of the air is not as high as some people think. I don't know what the exact figure is. I think it's more like 8 to 10% or something like that. And then the rest of it's, other inert gases,
It's about
nitrogen,
18, I think.
Something
Nitrogen
like that.
mostly, and a bit of
Nitrogen's-
CO2.
Yeah, and there's a tiny bit of things like helium.
I think there would
Tiny
be, probably.
elements like that. Yeah. So why is it toxic?
So it's intrinsically toxic because during the metabolism, there's radicals are formed. Have you heard of radicals before? So they are basically molecules are broken, and bonds in chemistry always want to be in pairs.
Right.
I'm not a chemist. I hope I'm not explaining this badly. So basically, the molecules are broken apart in a way that a single electron is sitting there, and one's desperate to find another electron to make a pair again. And during the whole oxygen metabolism, these molecules are formed, and then they're attacking DNA, proteins, and doing damage. So it's a complex one because we obviously need oxygen. So we need oxygen to survive, but in principle,
for energy cycles.
Yes, without oxygen, we couldn't get energy. Yeah, we would die very quickly, obviously.
Krebs cycle.
Electron transport chain is all part of the same ultimately. Yeah. So it's the final acceptor of the electrons that are flowing through when we're breaking down our food and doing all the redox oxidation reduction reactions. Then oxygen is the final one, and it turns into water, right?
But inherently, it is toxic.
Yes. So during that process, there are radicals formed. So I think it's really the whole toxicity thing is a fascinating topic in its own right. So we do need oxygen. We have obviously evolved to cope with the oxygen. Maybe if you're going right back to the start and think of the salt-loving bacteria, they obviously have coped also with something, and it's no bother for them anymore. I don't know.
Do they need the salt?
They don't really need the salt. So I was just thinking that's probably not really a good analogy, actually.
But at times
the archaea that are dependent on the salt, they basically need it now. They couldn't do without it anymore.
The archaea, very interesting.
The archaea that actually accumulate salt inside, they had to change their whole biochemistry to do that. So they adapted to that environment the same way as we evolutionary have adapted to, there's suddenly loads of oxygen. I don't know how many million or billion years ago. The plants made all the oxygen, the organisms adapted to the oxygen, right? So they made use of it, but at the same time, the biochemistry intrinsically is quite damaging, so they had to do something to counteract it. So our body is full of stress response mechanisms. So we have these antioxidants in us, vitamin C, for example, and they're there to mop up these radicals all the time. We also have enzymes, and these anaerobic bacteria in the gut don't have them, and also oxygen-tolerating bacteria have these enzymes that neutralize these radicals. Yeah, so there are enzymes that take away the radicals, so that action doesn't happen anymore, that cell constituents or molecules are damaged in the first place, but any damage that has been done, there will also be other systems cranked up that can repair the damage. So as long as you don't overdo it, and I'm not an expert on your breathing, so I don't know whether
it's making me feel good.
If it makes you feel good, I think that's fine. It should be fine.
Strong when I do it. But there's also some element, though, of a low level of cellular stress being a good thing.
I was going to...
Because it sort of triggers spring cleaning and... clearing out bad cells and repair. Repair and test. So,
and we are always exposed to toxins. So, the oxygen is a very simple example, but we're constantly in the environment potentially exposed to something. So if our stress responses, our stress coping mechanisms are sort of already there a little bit, induced, you would say, if they're not dormant, and the body has to react first if you're hit by this toxin, or this toxic substance, then your body can deal with it more quickly. And that's actually one theory how a lot of plant secondary molecules, these polyphenols, work. Yeah, we don't really know how they work. And how much is it an antioxidant effect? How much are actually these polyphenols cranking up our stress responses, or it may be something else altogether. So one theory is that basically they're actually slightly toxic for us. We see these things coming in and think...
I've not heard this one before.
Oh, really?
No, I've not heard this one before. I like this one.
Full disclosure, I'm not an expert in polyphenols. You need to go to some of my colleagues for that. But that's what I understand from my general reading, that it's one theory, and there are probably many different mechanisms in how these polyphenols work.
Including the microbiome chewing them up?
Well, So they're, as far as I understand, they're not really a major food or energy source for the microbiome, but the microbiome may have enzymes that convert them. So it's called biotransformation. It's not really, this is a food for me so much.
Because they like the fibre.
Yeah, they like the fibre.
We're going to talk about that too. Lots on fibre, yeah.
Yes. So yeah, one idea is that maybe it's actually a system, a mechanism to say, yeah, actually, this is potentially not good for me. The amount of food we're eating is cranking up these stress coping mechanisms, but not really making us ill. So it's a good thing. That's so interesting, isn't it? I know it is.
Because some of the most polyphenol dense foods are stressed plants.
Yeah.
Like the purple broccoli, for example.
Yeah.
So they're stressed, we're eating them, it's making us a little bit stressed.
In a way, yes.
In a way.
Yes, your biochemistry.
Is there some sort of deeper meaning there?
No, I think
we're going a bit off piece now on this podcast.
No, not at all.
I like to explore that a little bit.
But, it makes sense, isn't it? Because the plants do it as a coping mechanism to not be eaten by bugs, insects or whatever. So because it's humans. Toxic, And, we also have plants which we can't eat raw because it's too toxic for us. So when you then heat it up, you destroy that toxin. I'm not sure whether they're all polyphenols, but let's just say in general plant secondary compounds. It could be one way and I forget now what all these enzymes in the gut wall are called, but I think some of them are believed to latch onto these enzymes and trigger them to crank up these.
So does this mean if you eat too many polyphenols on plants, it might be a bad thing for you?
Again, I'm not an expert, but I suspect what we normally eat, the amounts we eat, is fine. Yeah, I'm not worried at all in eating plants.
Supplement heavily with polyphenols.
Oh,
yes.
So what sort of supplements are you taking?
Like fisetin, quercetin. I had resveratrol for a while, but these are flavonoids. These are polyphenols. I mean, quercetin's quite interesting because it has a number of potential medicinal benefits, which I don't require. Like, for example, for people with allergies, they anecdotally have very strong responses. Now, when we first met as a medical student trainee, I would have said, no, if there's not meta-analysis, RCT, I'm not interested because I'm training to be a doctor. But now I've moved on from the practice of medicine. I'm more open-minded. And I have actually got a view now, which is that I think if the anecdote doesn't match up with the data, and there's a lot of anecdote, then the data is just not there yet. That is a view I've taken. Because if you go to these medical conferences, you speak to a doctor, they say, we have our pyramid of evidence. And at the bottom is anecdote, and at the top is meta-analysis. And we're not really interested in this as an RCT, systematic review or meta-analysis. But actually, hold on a minute. Maybe it's just we've not done the studies yet, and that anecdote's real. So I believe people when they say, well, actually, my allergies get way better when I take quercetin. I believe that. And I think there's health benefits with flavonoids, but I'm going to look into this more now to establish if my extremely high dose of flavonoids are potentially having a damaging effect.
I can't say for sure it's not going to be good for you or it's even bad for you, but I wouldn't do it. So my philosophy is, I think along the lines with you, that if it says we don't have the evidence, I wouldn't say don't do it if I don't find negative evidence. But, one thought that is triggered by what you were just saying is, for a long time, and you still hear this now often in the television, in food programs or something, is, for example, about vitamin supplements.
Right.
Water-soluble vitamins, you can't do any harm. Just take them anyway. Your body will just get rid of them. And I think that's not really, there's too much evidence now to say that's too simple. It's not true. And I think vitamin C is an example, isn't it? Sports people use a lot of it. And I think the problem is they're actually messing with this whole stress system.
Yep.
Yes, they're completely messing up the regulation. And again, not my expertise. So, this is on very broad terms of this whole stress-inducing the mechanisms that help the body cope with stresses because it's flooded with vitamin C. And there could be different effects of all this. So I think one of my major microbiology professors in Germany said quite often, everything is a toxin. It depends on the concentration.
Love that.
Right?
That makes sense.
So I would be very careful.
Some stress is good. Some stress is good. A little bit of stress is almost always good if it's in the right setting.
Yeah.
But too much stress, whatever the trigger, is probably not a good thing.
It's not just the stress, it's really having too much of one molecule. So I give you another example of vitamin D. So I take very few supplements, but I do take vitamin D
yep.
because we're living so far north and all this.
Yep.
And then when
nice
I started...
sunny day today though, hey? It's a nice sunny day today.
Yeah, but in the winter we're not getting any, I'm afraid. So you need to wait till April here to get any benefit from the sun. The sun is here too weak to make vitamin D through your skin.
Yeah. Well, so can I just push back a little bit?
Yes.
It is weaker because it's not as high in the sky. The day is also shorter. But you could get your vitamin D if you lay out in your swimwear.
I don't think so.
Yeah, I think you can. I did a bit of work on this. It's not practical because you get too cold.
Okay.
Yeah, so you can't.
Yeah, I thought the energy level of the sun rays is so weak that it just doesn't trigger that reaction.
No,
That's what I read.
Yeah, but you should take vitamin D. I agree.
Yeah.
Yeah, and I do too.
So when I started doing this a few years ago, I took it with calcium and magnesium because what's good? Calcium, magnesium, you need them.
Yep.
So, and then I came across an article that found, and I can't remember the details. It's years ago, but it was a randomized study, I think. They found that there must have been an epidemiological study because they found people who had taken calcium supplements had a much higher rate of dementia.
Oh, gosh.
Was it dementia or heart disease? Anyway, it had to do with clogging up the arteries. Both linked.
Yeah.
So basically, the theory there is if you take your calcium in form of one pill, then you get such a massive influx of calcium very quickly into your body because that pill is, dissolving in your system very quickly. And that whole dose, which is often 2 or 3 or however many times daily recommended allowance, they're normally over concentrated. Don't understand why, but exactly. And then the calcium that would be a 1 daily allowance, which you get a little bit with your breakfast and a bit more with your lunch and a bit more with your dinner in a very complex meal where it will slowly trickle into your body. Suddenly you get maybe 2 or 3 days worth in like half an hour. And that people believe, I don't know whether that's ever been proven, but that was the hypothesis behind it, that you get such a spike of calcium that your arteries are getting clogged up because suddenly it's unphysiological concentrations. Yeah. So my philosophy is very much, so vitamin D, yes, I take fish oil because I don't really like fish. Yeah. And I have sort of starting arthritis issues and pain issues. So I think for me, that's something I need to take because I don't get it from the diet enough, because I don't want to force myself to eat oily fish twice a week. And I also take devil's claw. We can go back to that if you want later.
I'm very interested in devil's claw.
So devil's claw, I started getting these quite painful, horrible little knobbly knuckles a few years ago, and my mother had that very, very badly. So I just went, when I was visiting in Germany, went through the pharmacies and asked a few pharmacists, like, what could I do? Because whatever I had found in my reading, I wasn't really getting anywhere else that I could do because I already eat quite a healthy diet, I think. And one of them said, try this, and it's devil's claw. And I think there's quite good scientific evidence that it is anti-inflammatory and really counteracts inflammation, arthritis, both osteo and rheumatoid arthritis. And you know what? I started this 2 years ago or so.
You see benefits?
It stopped in its track. Pain is gone.
Amazing.
And, it's also gone back a bit. I think it's not as swollen anymore as it used to be.
That's amazing.
It seems it can interfere with your stomach, and I'm a bit worried because long-term usage could, nobody really knows, is it going to be okay in the studies, in the shorter-term studies, everything seems fine, but currently my benefit is so much.
That's incredible.
That
What is it? I mean, it's some sort of plant compound
it's a complete
or something?
plant. It's a plant. It's the complete plant extract. And again, there's one ingredient. You talked about, what's in turmeric?
Curcumin?
Curcumin, yeah. So, that's another one. So that would be the turmeric equivalent. So I take turmeric in my food, but I don't take curcumin because I believe after that calcium story and hearing about the vitamins and thinking we shouldn't just think bang it in, it's good for you. I mean, that's kind of unnatural, and I always think back what's physiologically normal for us. Yeah,
I'm going to show you after this recording my supplement pill box, and you're going to laugh a lot. It's full morning, lunch, and evening. And actually, honestly, the way you've just talked to me about all of that has made me deeply reconsider my strategy. I'm being 100% serious. I mean, really, really, really serious about that. I'm going to really think about my strategy because it just makes so much sense to me now that you've said it like that, that if you dump a whole load of a micronutrient into your body, you are going to achieve a concentration way higher than you would ever be able to achieve otherwise. Now, the flip side is, one of the benefits of supplements is you're able to do that. So vitamin D, for example, the EPA, DHA in your fish oil, you know, and the flavonoids. It's really hard to get high concentration of specific flavonoids. So there's a double-edged sword to all the supplementation, which is, yes, you can achieve supra-physiological, quote-unquote, through your diet, through supplementation.
Yeah.
But the flip is, as you've said, maybe it has an unintended consequence.
Yeah. Do you want that? Do you want more than would be normal for you? If you think back how many hundreds of thousands of years, how people lived, nobody would have been exposed to this. And, you know, I don't know, do you try to push your lifespan beyond what's normal? I just want to stay healthy. You know, I want to be as healthy as I can, and I'm worried about the potential consequences of overdosing something because my body is not really evolutionary. I don't think my body was ever expecting to be bombarded with stuff. And also, you know, think about it in these studies where they're looking at resveratrol, for example, and then say you need to drink 100 bottles of wine to get the dose you need to see the effects.
Right.
Well, you know, do you want to achieve that? Because it's not just going to be that one molecule. That's also why I take turmeric over curcumin, because the turmeric
I thought you were going to say that's why I drink 100 bottles of wine a week.
I wish my stomach wouldn't let me do that.
I was going to say, it better be good stuff if you're drinking that much,
But, you know, is it then still meaningful for keeping us healthy if you need a dose that is 1,000-fold over what you could ever eat? And if you think for somebody like you, you must take a lot of secondary plant compounds in already. So you have a huge cocktail. And some of them will obviously do exactly the same as your curcumin or your quercetin or whatever do, you know?
I mean, every day I'm eating orange, grapefruit, blueberry, raspberry, banana, apple, a green supplement, several different types of vegetable, artichoke, pepper, sometimes broccoli, asparagus, every day. Mushrooms, every day, every day. Well, one of the perks that antibiotics, although I have to say, and if any of you are listening, this is not a critique, not that many people take up all the perks, I have to say. So we have the Fresh Pest green juice, we had for a while greens powder, we have unlimited fruit and veg, so it's all there. And I just kind of eat a lot of nuts as well. So maybe I don't need the quercetin every day. So I'm going to think about that carefully. But just to push back a little bit, I suppose, because that's my job as the host. Could you argue that we understand biology now better than we ever have done, and it may be advantageous to get a higher concentration of something than you'd typically be able to get through diet? Or is it always going to be about the body's evolved in tandem with availability of food and quote-unquote normality, and the optimal strategy for your health is to try and live almost like a hunter-gatherer in some respects?
I think that the second is my thinking, or I think I would call it philosophy. I'm not saying as a scientist now I could tell you it's definitely you should do this and not that. I'm not sure, you know. But just from the philosophy, from the concept, it makes sense to me to say, well, you know, kind of what is normal for us from an evolutionary perspective. How our bodies evolved with its environment. And then we can push that a bit, I think, you know. But in the bigger scheme of things, for me personally, that's all I'm aspiring to as well. I'm not one of these people who want to live 200 years, you know. I think I'm quite content with the idea that we all have a limited time span. I may change my mind when I'm 80, or if I make it to 80, I don't know. You know what I mean?
Call me at 80 and go, James, give me the protocol for breathing and the ice bath.
Too late.
I want to go to the next level.
Too late, Petra. You should have started when you were.
But that's my philosophy. My philosophy is the sooner you start, the longer. So there's lifespan, which is I was born and I die. Delta, lifespan. Then there's healthspan, which is whatever proportion of the delta of your lifespan you're actually healthy. And healthy is individual. So I know guys who want to just be as muscly as possible. And you wouldn't believe the extremes.
Oh, I know.
They're
I know some of them.
They're injecting themselves with insulin,
Yeah.
growth hormone, and a cocktail of synthetic and natural hormones every day. And I've had a couple on the podcast, actually. Well, one guy on the podcast who's brilliant. And he will say things like, well, actually, there's only 1 week of the whole year where I actually feel good. I'm like, sorry, what? And it's because for 6 months of the year, he's force feeding himself. And for 6 months of the year, he's starving himself. Well, and there's a week in between. And I was like, that's really weird. But that's what he wants to do. But I think what you're describing is I just want to live a healthy, happy life. And I don't need to be 100, 150, 200.
Yeah. So in terms of lifespan, I'm not too worried to make become the oldest lady in Britain in so many decades. Health span, I do want to stay healthy as much as possible. But also, to be honest, I'm worried about, as we talked already about, what harm could I do because I'm ignorant to certain things. But also, how important is it really? I know some people who lived very unhealthy lives and made it to 90 or 100. I know some other people who are super healthy, slim, everything like this. Every box is ticked, and they still ended up with early onset dementia in their early 50s.
You see these videos of, so what's the secret? Just smoke.
My granny smoked into her way into her 70s, and she died in her late 80s. Like she was smoking probably 2 packets of cigarettes a day or something like this. Understand. So there's obviously so many facets. It's our genetics as well, and all sorts of things coming together. And for me personally, I'm not putting this either lifespan or health span over everything else. I want to have fun as well. I do sometimes drink too much, and I sometimes do whatever I like and don't get my sleep and all this, because I also have learned, I think, over recent years that my psychological state has a real impact on my physiology. So I think almost being happy and not too stressed maybe about have taken all my supplements today, but also keep you healthy, because I do think that happiness is half the battle in staying healthy. And
100%.
I have definitely had sort of physical effects from stress, and they have resolved through the pandemic because suddenly the whole work pattern had changed, and that's was one person making an observation. But I think I have learned for myself how...
So your stress levels are better now than they've been in the past?
Yeah, so I had constant stomach problems, I've been actually on omeprazole for like 6 months, and I said in the end to the GP, that's all they were going to offering me. I said, and it didn't even help that much. I said, thanks for that. It was all stress. And then being able to work from home, and I have no problems with my colleagues, all fine, but the work environment, shared offices and all this, I'm one of these strangely very easily distracted people. I can't shut off. I was really stressed out, and
so it helped you working from home.
And it took like a year or two, and suddenly one day I realized this is the first time in years, and I haven't had any stomach tweaks, and I can't go out for meals in the evening anymore. I couldn't go to a restaurant or anything. I was, my stomach was completely done in. I really expected that I would end up with an ulcer, or maybe I was on the way to it, and it's completely resolved.
Wow.
And I'm pretty sure it was due to one very stressful aspect of my work having been taken out on a pretty much permanent basis. Yeah. And I found
Are you still working from home now, or are you back in the office?
I'm mostly working from home. Luckily, my employer lets me do that.
That's excellent.
And I walk 20 minutes, so I go for all meetings and everything, do everything in person. But I can actually now sit in my home office and concentrate and not be distracted anymore. So it made a huge difference to me.
That's really interesting. I'm trying to get my people back to the office, but again, you've made me reframe my perspective a little bit, because, I think when you're the CEO, you're first of all, you're stressed all the time. And there's a number of different reasons for that. One, you're accountable for everything. 2, the buck stops with you, in line with the first thing. But any delay or any issue, you're going to have to report it and take the hit. And when it's going well, it's the best job in the world. It's the best job in the world, full stop. But one of the other things that people don't realize is you're actually the kind of epicenter or the peak of all the problems within the company, which filter up and escalate. So essentially, at any given time, have the summation of all the worst problems in the company every day. That's really stressful, right?
Yep.
And one of the defaults, I think, when you're CEO and you're stressed is, well, where's everybody? You know, where they're not in the office. So maybe I need to reconsider that a little bit as well. What you've said is very profound. It's essentially working from home has impacted your health in a positive way, and that's probably improved your work output as well. Almost certainly, right? Now, on the subject of your work, because we've talked about supplements and health and everything, which is great because we like to cover that in the podcast. What are you focused on at the moment?
So,
and I do also want to talk about fibre, by the way, because...
Yeah, I was going to talk about fibre now, if that's okay.
Yes.
So yeah, my main...
And by the way, one question, sorry, before you go. Was Harry Flint your supervisor or was he a colleague?
No, he was my line manager, I would say. So
he brought you to the Rowett,
came to the Rowett in a tenure track position, so I worked in his group and worked very closely with him for many years. And I was very lucky because it was very enjoyable. And I think we were very compatible in our thinking and all this. So, you know, I'm one of these details people and all this, and he's the big ideas guy. So I think we worked very well together, you know, and then we would keep each other on the straight and narrow sort of stuff, you know. So,
of course, he came on. He's probably the most prepared person I've ever had on the podcast.
Prepared?
Yeah.
In what way?
He came with like a whole pile of notes and plans and things. Yeah, you know, we should maybe talk about this. And if you listen to the episode, it's quite funny because I would love to have him on again, actually, but he sort of sometimes was like, well, I just need to finish this point and to finish off the story. And I just loved that because you could tell he was getting really into it. And I don't think he'd been on a podcast before. So he'd sort of had everything well organised and wanted to tell lots of stories. So I thoroughly enjoyed it. But he'd retired, I think, just as I was getting started with enterobiotics. He made some recommendations. He I think via you actually, said I should speak to Ailsa Hart. That was Harry. So, you know, and I did go speak to Ailsa Hart. So back to you and your work and fibre. So yeah, when I came to the Rowett, I worked in Harry's group, and that was early 2000s, and he had a previous PhD student who had isolated all these butyrate-producing bacteria. So there is some of the bacteria in the human large intestine. They make this compound called butyrate, and it's health-promoting. It's really good for you. It is anti-inflammatory. It counteracts cancer in the gut. It gives us energy. It's actually the main energy source for the large intestinal wall. So it really ensures that the colon wall is working, that it is nice and tight and doesn't have holes in it, that all these toxic things that will be in the lumen can get in. And then also it's taken up and together with other compounds, so-called short-chain fatty acids, it also goes into the body and regulates all sorts of different things like glucose metabolism, appetite, and so on and so forth. So there had been a lot of interest in it, but very few of the bacteria were known that actually makes this magic compound butyrate. And then the strain collection was literally there or being built up. And then I came on board, and with my very molecular background, my main work area really was to work out the biochemistry behind butyrate metabolism. We kind of knew how they make it, but we didn't know the details. So that's very much what I worked on in my maybe the first 10 years or so. And then now I'm still interested in these short-chain fatty acids, but I'm also looking much more on the other side of things. So if you think very broadly about fiber metabolism, fiber being those plant compounds that are not digestible in our upper intestine. So they reach the colon where they are then fermented by the bacteria. That's a huge group of different types of molecules. And so in my early years, they're all broken down to sugars, essentially. Let's say glucose is a very simple sugar molecule. And then in the first years, I worked from starting with the sugar and looking what happens then once the bacteria have the sugar and making these short-chain fatty acids. So I focused on that metabolism. And now that's obviously part of the whole deal, but I'm much more interested now in all these different types of fiber, both in their chemical composition, but also their physicochemical composition. So if you think of fiber, it's mainly carbohydrates. So
consisting of monosaccharides or sugars, and there are many different types of sugars. There's glucose, fructose, arabinose, xylose, and so on and so forth, galactose. And then in the plant, they're linked in different long chain molecules. So the plant cell wall, so of your fruit and vegetables, cereals as well, is made up of these long chains of these sugar molecules, monosaccharides, to make this really rigid cell wall for the plant. Mm-hmm. And there are many different types of molecules. There's cellulose, which is just a linear chain Classical of glucose here. Then there are arabinoxylans, which is arabinose and xylose molecules, and so on. Some of them are linear, some of them are branched. How they're bound together, the monosaccharides differ. So it's a huge complexity of different molecules coming in. And we just don't have the enzymes in our small intestine to turn them into sugars. So this is why they're food for the bacteria. They're passing right through. And then different bacteria contain different enzymes coded in their genomes to break these different molecules down into their monosaccharides. And then the monosaccharides are fermented to starch and fatty acids.
So at the most basic level then, the fibres are complicated sugars, complicated carbohydrates, at the most basic level. And some complicated carbohydrates we can break down in our upper GI tract.
Starch is, we can break that down in principle. So starch is another macromolecule that consists of glucose, but it is in a different bond configuration to cellulose. So it just is three-dimensionally, it's different. And it needs different enzymes to break them down into sugar. And we don't have the enzyme for the cellulose bond, but we have it for the starch bond. So principally, we can break starch down in the small intestine, but often we don't break all of it down. Because there's only so much time the food moves through the system and the enzyme is chipping away, but the starch is often, it's a storage molecule for plants. So it's often big globules. And the enzyme is nipping away at the outside and is liberating some glucose, but some of that big molecule is passing through, and then it becomes food for the bacteria as well. And that fraction of the starch which is passing through is called resistant starch.
Which is a hot topic right now in the press and so on and so forth. People talk about get your resistant starch in. Yeah. So how do we sort of summarize fibre then before we jump into the details around different types, for the listener, because everyone hears, well, you need to eat more fibre for your microbiome. But it sounds like it's a little bit more complicated than that. Maybe you want different types of fibre. So what would be the kind of summary then that a layperson could take away from what we know about fibre?
So I think what's also in the press a lot is eat a diversity of different fruit and vegetables. And that taps very much into the fiber side of things, also the polyphenols and everything else. It's not just about the fiber, but a nice mix of different food ingredients gives you the different types of fiber.
Got it.
And different bacteria can consume the different types of fiber. So there's a very complex metabolic network of the microbes in the large intestine. Some of them can break down starch. Others can maybe nibble a little bit away at the cellulose in plant cell walls, yet other ones do other things. So there's these primary degraders that can attack the macromolecules and break them down into smaller units. And then there's a complex web of other bacteria that then cross-feed from that. So if you take a wide variety of fiber, you're more likely to stimulate many bacteria, keep a nice, diverse ecosystem down there. And that applies to ecology generally, not just in the gut, but in general, a more diverse ecosystem tends to be a more resilient ecosystem. So if you compare, a monoculture in a field, if there's one pest coming in, it can wipe them all out. If you have a very diverse ecosystem, like a forest or something, it's much harder for a pest coming in and wiping out the forest. So the analogy broadly probably holds for the microbes in the large intestine as well. So, there has been a while in the media, I think is what's happening now in the television programs has got much better. But I remember one program about 10, 15 years ago where it was all about how do you improve your diet? And this was people eating a very fiber-poor diet. And then the presenter was it's very simple. You just sprinkle some of this white powder and everything will be fine.
Wow.
And that was inulin or resistant starch or something. And, so the message was given, you can eat your burgers or whatever, and then you just sprinkle something on top.
I wish it was that easy.
I don't think it's that good, because these tend to be refined. It's just one type of molecule that goes down there, and once it reaches the start of the colon, it's so fermentable. There's an explosion of bacterial activity. It's maybe quite okay in the proximal colon, but the stuff in the colon is passing around for quite a long time, and then there will not be much to eat for the microbes further down. They can't make their health-promoting butyrate and short-chain fatty acids. So it's not a good idea. And also, obviously, if you use masses of one type of fiber rather than a nice fiber mix, you don't preserve the diversity.
And what about the difference between soluble and insoluble fiber then? What are the different ways of classifying fiber for the listener to learn from?
Yeah, so there is soluble fiber where the molecules basically get solubilized into the water that's in the system as well. And then there's insoluble fiber. And they both play a role. Again, there will be different microbes that can use different types of fiber. So you want a mix of both of them. Some of the soluble fiber also have other effects. So we should also think about these in terms of health. So fiber can also have water holding capacities. It can change the viscosity of your food. So it may also make certain foods stay in your stomach for longer, because your whole system, your whole digestive system is obviously influenced by the consistency of it as well. And then there's the insoluble fiber. So some of that insoluble fiber will just pass through. It will not all be degraded. So if you look in a fecal sample, you will find fibrous matter. And again, it can have physical consequences. It can hold water and change viscosity and lead to bulking. So if there's more material in the gut wall sees there's something there and maybe does more muscle movement to move the material on. So there are all sorts of physical factors. But also from the microbial perspective, I think we need to do much more work on that, because we don't understand in detail. And that's very much what I'm interested in, the insoluble fiber structure. So the plant cell wall fiber, it's all these different molecules still bound together. And if you chew a carrot, you will have bits of carrot floating down there, right? And very few in the field have actually done work on this, because it's very hard to study. We have all taken from the companies a purified fiber where we just have one type of molecule and we see how bacteria convert that, but very few of us have really looked at these complex food particles, basically. And now in the last few years...
what impact they have on the microbiome.
Yeah, how important is it? So we talked about this white powder, which we know is very purified, goes quickly through, but if you don't take any supplements, fiber supplements, but you eat only plants, how big is the effect on your gut bacteria, on your health? I don't think we have. No, we don't know that. That's what I'm saying. Very few of us have worked on this because technically it's much harder to work with this. As a microbiologist, you want nice, clear solutions, media. So then when the bacteria multiply, you can do a measurement called optical density. If it gets cloudy, your bacteria have grown. Imagine insoluble fiber. Right? It's already cloudy.
We did some work together in that project. We can't go into all the details because a lot of it's confidential, but we did some work together on complexity of carbohydrate. I was reading that before we came in, actually. So what do we know about fibre then and its importance for the gut microbiome?
Well, we're currently working on that, and other groups now, I would say in the last 5 to 10 years, more and more groups are tackling it, because like I say, it's technically so much harder to work with a very complex insoluble material, which is obviously much less characterized. But basically, we need to understand this better, because there are other functions in the microbiota that may really be influenced by this. So, for example, how well can they persist in the large intestine? You will be aware that there are different microniches in the large intestine. We say there's the lumen, so bacteria that are swimming around in the soup, essentially, and there's the gut wall, where bacteria may be attached. But there's another surface. There are plant fibers. Yeah, well, if you don't eat any plants, then you won't have very much of that. But if you just eat a carrot and you've chewed it, you still have fibers. So if you look under the microscope in a fiber in a fecal sample, you will see 3-dimensional big fat particles. They're much, much bigger than bacterial cells, right? And they're essentially a colonization surface. So in nature, microbes make biofilms. You find them in lake sediments and in soils. So where there are surfaces, microbial communities will attach. Some bacteria have adhesion factors and mix slimes and all sorts of things and sit on that surface. So in the gut surfaces, they do that on the gut wall. But
some people are going to be so freaked out hearing that there's slime in their body. What? No, I don't want any.
Well, there's quite some slime on your gut wall.
Yeah, no, I know. For sure. There's lots of mucus and slime in there.
Absolutely.
Yeah.
So, we don't, at this moment in time, really have the scientific evidence in detail, but common sense tells me it makes physiologically a huge difference if you eat
100%.
whole foods and have these big surfaces, and then you have these colonies forming. And as I touched on earlier, the bacteria cross-feed to each other. So there will be some bacteria that even have little docking domains. They can dock onto the cellulose. They have little protein domains that can latch onto the...
So they're adapted to attach to cellulose?
Yeah, absolutely. So cellulose degraders tend to have carbohydrate binding domains, which are specific for cellulose. Wow. Starch degraders have carbohydrate binding domains that are specific binding onto starch. So they need to bind onto this stuff to be physically close enough, And then these bacteria may start breaking down the cellulose. And these macromolecules are so big that they need to do all this outside. Yeah, they can't take the cellulose in because it's massive compared to the bacterium. So they break them down into shorter units, just a few sugars together. And then they consume some of them, but some of it is normally diffusing away. Okay. So another bacterium may benefit from it. So now... how important is that biofilm structure to facilitate these cross-feeding reactions? Because that happens on very small distances. If you're too far away from that cellulose degrader, you're not going to get any, right? So do bacteria have a benefit that can also then say, I can latch onto this biofilm? Not with that domain, because they may not have it, but in another way or form. So we really don't understand this to sufficient detail. And it is hard to study. So we're working on this... we have also looking at the carbohydrate composition of different plants. And again, other people have done this. So, your cereals are different from a compositional point from your fruit and vegetables. But then especially within the fruit and vegetables, you have a lot of variety. So we did a study with a recent PhD student where he took different vegetables. What was it now? I think it was carrot, kale, apple, beetroot and beet leaves. I think it was 5. And he extracted the plant cell walls from all these different plants. And then we did, in the lab, we did incubations with fecal samples to see, and he characterized what molecules, what monosaccharides are actually in there. And what we found, for example, there's a component in fiber that's pectin. Yep. And the pectin from the beets.
Also found in apples?
Yes, yes. It's really found to a degree in most fruit and vegetables, not so much in cereals. And we found that the carrot pectin really stimulated Eubacterium rectale, butyrate-producing bacterium. But the beet pectins, and that was both the beet roots and the beet leaves, they stimulated Bacteroides much more.
Interesting.
And then we looked at the composition.
Do we understand why? Molecularly, cellularly?
Well, the thing is, when we looked at the monosaccharides, so the sugar composition of the pectin fraction, it was really high in arabinose in the beets, and it was really high in galactose in the carrot.
Different sugars.
Yeah, different sugars. So we expected, obviously, that the composition is somewhat different. Right. But they were the two extremes. The other 3 were sort of in between. But I never expected that the pectin composition would be so different. So that also goes into eat different foods because it will stimulate different.
So all of this dietary advice, eat your fruit and veg, has kind of been consistent in many respects for a long time. Eat real foods, eat your 5 fruit or veg a day. And we've known that this is associated with health benefits. Could it be that it's having a direct impact on the microbiome because of the carbohydrate, and the impact on the microbiome is having an impact on the wider body? Potentially?
Yeah, absolutely.
So then that leads me to the next question, which is how do we optimize our fibre intake for our microbiome, gut health, and then our own health? What do we need to do?
Well,
Easy question for you to answer.
No, I don't think I have an answer to that. So for me, going slightly back to where we were earlier as well, I think it is, again, a mix of, I don't get too stressed about 5 fruit and veg a day or 30 different plants per week. I think these things are useful. I'm not rubbishing them, but I think for the 5 fruit and veg, we know that it should really be more than that, I think. But we thought better give people something that they can aspire to, because if you tell them eat 8 or 10, they're probably not going to do anything and give up, you know? So, they're just to help people to have a target, but the targets are not always in line with what we as scientists know should be done. So I think they're all useful. It's useful for people to say you should try to eat 30. But at the same time, my personal philosophy comes in again. I want to enjoy life as well. And also, I don't want to get so obsessed with something. I don't want to constantly think, oh, is this okay today? Oh, am I eating something unhealthy? And I saw it with my dad. My dad had a sort of wake-up call in his late 50s. And since then, he's really quite stressed when he's just eating that cake at a party or something.
Right.
And that's a bit of a shame. You should allow yourself to, enjoy. Because, again, my philosophy is maybe I'm making that up because it's so handy to have that philosophy. It makes me happy, and as long as it's within reason. I think the problem really comes if people eat unhealthy all the time. Then you have a huge problem. Yeah. If you generally eat a healthy diet, it's fine. Also, again, from a personal perspective, I grew up on an orchard, but I hate a lot of fruit. I can't stand it. I can't stand it.
But there are people listening to this. I know that they listen because they tell me and they write to me that are, I suppose, in some respects, like me, trying to optimize and get quite geeky and scientific about everything. And I think that the story on probiotics, I think, is quite clear. What you can buy at the moment off the shelf, it's probably not worthwhile if you're healthy. It might be worthwhile if you've got traveler's diarrhea or you've had some antibiotics, just classic examples. The prebiotic side and the polyphenol side is quite interesting because, in many respects, polyphenol-containing foods are prebiotic because they've got fibres in them as well.
Yeah, if it's not a polyphenol supplement, I guess.
Right. And then prebiotics more generally, they could be useful for some people in a supplement format. But if you eat all your vegetables, then you should be getting prebiotics within your vegetables. Is that correct?
Well, maybe we should talk
push back on any of that.
A little bit on prebiotics and fibre. Yeah. So there is a lot of confusion in this, I find. And I personally,
let's talk about that.
Again, I'm not too hung up on what we call it in the end, but there's also danger that people then call everything. So,
fibre
like
prebiotic, which is not sure.
Well, I mean, the prebiotic definition is changing, right? It changed over time, and I'm pretty sure it will change again. So just to summarize, maybe a dietary fibre, as far as I understand, is what I said earlier. It's your plant food that goes through your system and is not digested further up. So strictly speaking, that could also contain some resistant protein. So we can digest protein in the upper gut, but there could be some going through. I don't think you would class that as dietary fiber because it's dietary fiber, not protein, okay? So there's also resistant protein. But in the broadest sense, dietary fiber is not linked to a health effect as such. Yeah? It is just a very simple and clean definition. I think also needs to be 3 monosaccharides at least. So a disaccharide that may also go through is currently not classed as a dietary fiber, but we now get into the nitty-gritty, and I'm not really interested. It's about the concepts and the understanding, isn't it? So dietary fiber doesn't have to have a health effect, but we know, in fact, probably all dietary fiber has a health effect in some way or form. I talked about also the physical effects, the bulking, the water holding capacity, and so on. A prebiotic, by definition right now, needs to have a health outcome in the body, and that needs to be proven, kind of. And then also, I think still one valid, but maybe a slightly earlier definition was it had to be selectively fermented by a microbe in the gut. Yeah, so in 20 years ago, selectively means, so 20 years ago, when there was much less knowledge about the microbiota and its metabolism of dietary fiber and so on, when we very much thought the gut was full of bifidobacteria and not much else. Yeah,
Was that true at one point in time?
I think people thought that before all, like Harry Flint's and other people's work came up, and said, look, there's all these other bacteria here, because when people, before molecular techniques, mostly plated out fecal samples to see which bacteria are there, they came up with 70% bifids, because a lot of the other bugs didn't grow on their media under their conditions, or it wasn't anaerobic enough or whatever. So then I think the idea was like inulin is your classic prebiotic, stimulates bifidobacteria, end of story. And, our group and others then said, look, it's more complicated. Guess what? They are non-bifidobacteria that can also consume inulin. So that selectivity concept was the central concept of prebiotics, 15 years ago or so.
Right.
And then there was a lot of pushback, and now that's kind of going by the wayside. But I think at present, at least for one of the currently still valid definitions, it says it needs to be fermented.
Yep.
Yeah.
Fermented means eaten?
Eaten by the bacteria. And for me, fermentation means the bacteria take that fiber, turn it into the sugar, and then ferment the sugar down to these short-chain fatty acids. That's, as a microbiologist, my understanding of fermentation.
One question. Is the end stage of sugar digestion and processing in microorganisms always short-chain fatty acids?
It doesn't have to be, but in the gut it mostly is.
Got it.
Yeah. I think, like I say, there are different organizations out there and different definitions, but I think fermentation is still one tick box as a prebiotic. It needs to be fermented, and not all dietary fiber is fermented. So the cellulose, for example, or fractions of other ones may not be fermented, but they may still have a health benefit, but it doesn't have to be fermented for that health benefit. On the other hand, I think some people, I wouldn't class polyphenols as prebiotics if we're going by the fermentation definition. So the fermentation process is really that sugar is the main, or is a major energy source, and also building blocks for the microbe to grow with. Yeah, so that's its main metabolism. That's where it gets its energy and its cell material from. Yep. Whereas what they do with the polyphenols are little side reactions. They may be involved in that somewhere. Maybe they use them to dump some electrons in the whole process, but they're not the main food source. So in that sense, if you go by the classic prebiotic definition, polyphenols, I think, are not prebiotics. So
got it.
So in the bigger scheme of things, I'm not too hung up on definitions. I'm always more interested in thinking about the concepts and the functions and all this, but it's obviously dangerous that people can jump onto definitions and then everything gets messy.
Just listening to what you said, is it fair to say, as a summary, I hope I've listened correctly, that all prebiotics are fibres, but not all fibres are prebiotic?
Yes, I think you could see it that way, but then you may now find another prebiotic definition somewhere that's maybe not quite in line. But yes, principally speaking, I think for a food company to say this has a prebiotic in it, you need to have that health claim, and also due to some definitions at least, it needs to be selective, although that becomes less important, it needs to be fermented. Okay. But I think also the scientific field is aware of this, and this definition every few years is moving with the time.
Changes,
so I think it's just important for people to know this is not written in stone, because as we build up the knowledge, also our understanding changes, and therefore the definition may change. So, for example, that selectivity criterion, which was very central, let's say 10 years ago, 15 years ago, now most in the field say, we know it's not that selective. To me personally, is it that important that it's selective? And I think there is no one definition out where it says it needs to have a health benefit. So I think in one of these organizations has it even taken out completely. So dietary fiber is basically looking at it much more from a physical point of view and a human body physiology point of view. Where is it ending up in the body? Is it degraded up there or not? Whereas a prebiotic has this whole health claim associated with it. But often prebiotics are really dietary fiber, but they tend to be extracted. So if you eat an onion, you will eat some fructans. They are part of the natural matrix of the plant, of the onion. But then a company may take the onions and normally take chicory. But,
chicory is good, right?
Chicory has loads of fructans in it as well. And they extract just the fructans. And then you have that prebiotic supplement where you can put just the fructan on top. And my big research area is, can we really say that fructan is as healthy as eating loads of chicory and onions and wheat and whatever else. So I think I try to find the scientific, or to gather the scientific evidence together with many other researchers to convince people to say, don't just sprinkle some white stuff on your food, have a wide variety.
And are we there yet? Is the evidence base strong enough to say, look, you can buy inulin powder, you can buy pectin powder, you can buy chicory root extract?
So what you could do, I think the consensus now is very much don't just eat tons of fructans and nothing else. I think the consensus is there. It should be a variety of different fibers. Whether it's okay to make that up by now having a new white powder that actually contains 10 different things, or you want that whole food, I don't think we have enough hard evidence. But my gut feeling tells me take the whole foods, because again, you have the micronutrients, you have the polyphenols,
yes,
you have the food matrix, and that's something that we can maybe talk about a little bit more. We touched on the biofilms and all this. How important is that? And then we come to the next level of, should it be raw or cooked? Should it be a fine flour, or should you grate things finely? Should you eat them as raw, that you get big particles in as much as you can? For your grains, whole grains, as in ideally not into flour, well, they may just go through. It may not be the best thing,
Can we start to unpack a bit of that then? So
what I'm saying is...
Stuff. So what about cooking? Cooking versus raw?
Well, I think, again, from my common sense perspective, I quite like a raw carrot, but I would also not say no to a cooked carrot. I think a bit of both is probably good. Because going back to the carrots, if you chew a raw carrot, and again, I'm not a food structure specialist, that's from my reading only, but I understand the raw material, and you can get that feel when you bite into it. You break open the plant cell. Yeah, so there will obviously be particles where there's intact plant cells, but wherever you have the break points, big crack, and the cells literally come apart. So all that material that's inside the cell flows out, obviously.
Could be good stuff.
Could be good stuff. Maybe that's what would get rid of when you cook something and throw away the water later on. So that's one thing. Also, the fiber structure is then already different. When you cook a carrot, that pectin we talked about earlier is dissolved, that disappears. You may end up with eating it still if you keep the juices, if you cook it in a stir fry, and the pectin may still be in there, but it's now physically a totally different structure, right? So we don't understand that, and several of us are now working on this, and we're using all sorts of different things. I'm trying to set up microscopy techniques so that I can look at the fiber structure and the bacteria on top and all that sort of stuff. How much does it really matter? But my knowledge tells me it must have an impact. Whether it's a small impact that doesn't really matter, or whether it's a huge impact, I can't really tell you at this moment in time. But we're working on this. We're looking at, and other labs do this. If you work with flours, like grain flours, should it be coarse? Should it be fine? Maybe a mix of everything. What is the size of the flour particle? The surface, that's something I'm working on, the surface. Depending on how you grind your flour, you could have a really closed, dense surface, which the bacteria may find really hard to attach to. Okay. Or you maybe have an open, fluffy surface, which is a much better colonization site for the microbes. Does that affect how these biofilms are forming? And we're really only at the start of this. We don't know enough about it. So my hypothesis is variety, big and small and cooked and raw and all this. Variety. It again gives you the diversity that we already talked about in chemical terms. It makes sense chemically that you want the different types of monosaccharides, the different types of bonds. So it would also make sense on a physicochemical and food processing point of view to have a variety, because probably different ones are good for different bacteria.
So where does all of this go then in 5 or 10 years or 20 years or 50 years? If you're starting to understand the impact of cooking versus not cooking and complex carbohydrates and mixes of different fibres, like where do we go?
I think ultimately we need to look at all these different things together, and I even want to throw back in sort of my likes and dislikes. I'm not allergic to anything, but I really can't stand some fruit. I love berries. I eat berries every day. And then I can grade fruit into the sort of love it to absolutely hate it. And it's been like this all my life.
It, just out of interest?
Well, the story goes I couldn't
Durian
walk or talk,
fruit?
and I was refusing to get any banana stuff shoved into me.
Oh, really?
Bananas, the smell. Apples, can't really stand them. There's a story. I once came to work when I was a PhD student in Germany, and we walked along a long corridor and another long corridor and through 3 doors and all this. So I could smell the apple smell like from the very start coming into the building. So I walk into the lab and say to my mate, "You've been eating an apple, haven't you?"
Wow. You got
he said,
very whole
yes,
century.
But that was half an hour ago.
Put the apples away. Petra's coming.
So, I've tried all foods that I don't like as an adult again, and some of them I don't try anymore because I know it is something in my body. My body says, I really find this revolting, don't eat it, right? And I'm not forcing anything into my body that my body tells me, no, I don't like this,
so
because I don't think it's, I don't know what is. I don't know. I had this all my life. And like I said, I grew up on an orchard. I was exposed to this stuff all the time, So there's a certain amount of habituation, obviously. Some foods you don't like, but if you keep eating them, it's okay, But I think some of them are so extreme. And I wonder, is there actually something in my genome,
a banana,
that tells me maybe for me it's not good, And also generally, I'm not a very sweet person. I don't need loads of sweet things, and fruit tend to be quite sweet as well. Maybe for my whatever glucose metabolism, it's actually not the best thing if I would eat tons of fruit. And I love every vegetable on the planet, so I eat loads of vegetables. So I think people shouldn't be, and that's again only my personal sort of looking at like everything. My personal philosophy is, if you really dislike something, don't force yourself. If you think you dislike something, but, like people who drink tea or coffee with loads of sugar, that's normally not a dislike or a like. That's just a habituation.
This is a sugar addiction.
Yes. Yeah. But, there are certain things that you need to habituate yourself, but very quickly your taste receptors obviously change. And I've done all these tests, so I know that in my case it's really something much more fundamental.
That's fascinating.
So, and I think why should people force themselves to do something that probably again psychologically is not good for them? If you eat something you find disgusting all the time because you think it's good for you, it can't be good for you, right? So I think yes, variety, but don't stress about every last. You need to have all the different vegetables and fruits on the planet. I don't really think that matters.
And what about the sort of translational aspects of your work? Are you going to create new products, new dietary recommendations? Where do you see it all going and fitting together with regards to the future?
Yeah, so I do like the fundamental knowledge aspects, but obviously, and also working in the Rowett Institute, we're very much working with the food industry, for example, to get to this next generation of products. If we're saying, for example, we do want to create prebiotics or dietary fiber ingredients that are working for people, I also think we shouldn't be religious about, the only way to go is that full plant-based diet. I think it's also unrealistic. Let's see where the world is going. So I think it's okay if people really struggle with it, then we should find these new products. And the Rowett does a lot of this. I'm maybe not that involved with it personally very much, but, have your burger, but put some plant-based material in to make it more healthy and stuff like this. But I think where we're ultimately, as the whole research community need to go, we need to put all these different aspects together. It is not just the microbiome. It's not just the diet. It's not just your genetics or your immune system, which obviously is primed by your genetics. It's the whole thing together. And every of these systems is so complex. And I don't think we're going to be there in 5 years. But yes, personalization. And, it's exciting, these studies where, people now run around with glucose monitors. And everybody's reacting totally differently. And the microbiome definitely seems to play a role in this. But I think, again, host genetics will as well. And probably the state you're in that day, as well, obviously, what have you done yesterday and all that sort of thing. But I think that's really exciting that everything is coming together now. And over the last 10, 15 years, there has been such an explosion in methodology, and everything's going exponential, which is really hard to keep up with sometimes. I don't know how to read all these papers.
How do you keep up with it?
I can't. I find it quite overwhelming.
Alan said the same thing. It's not possible anymore.
No, it's not possible
So
anymore.
what do you do? Read the highest impact journals? Because
I
sometimes you might
tend
miss
to
some
do
gems.
my reading now really specifically. When I write either my own work up, I then read intensively about it. I keep a sort of tab on, I have a search going on PubMed. So I basically, every Monday, I get my alerts and I look through this for 10, 15 minutes, and everything that I think, this could be, if it's super, super important, I will download the paper right away. Otherwise, I just put the abstract into a Word file, and then I have a searchable database. So then I go back. If I have an invitation to write a review, I can go back to that and through the Word file very easily dig out those in the last year or two that came that are relevant. And obviously I use databases, but it's a bit of everything. I use the databases always to see what's new that I have maybe have missed.
Do you use AI?
No.
Not yet.
Not yet. I don't. I'm not a big fan, but I'm sitting on the fence right now.
Interesting.
But I'm actually finding it quite scary from a more societal point of view. Yeah. And also as a lecturer or teacher, it's very difficult to do student exams, and it's not my thing, but I also think it could, for certain things, I think humanity needs it. So I'm not really opposed to it in principle, but for what I'm doing, I don't see the application, and also I find it quite scary what could come out of it.
Oh, it's so powerful. Mm-hmm. It's unbelievably powerful.
Yeah.
I think this podcast will become an AI resource in the future.
Right.
Because what we're doing with this podcast, I think, is creating the biggest, most in-depth library of long-form conversations about the microbiome.
Right.
I don't think it exists anywhere else. So maybe the computers will be able to piece it all together and go, we listened to Harry talk about this and Petra talk about that, and as a result, we've got this new theory. Maybe, I don't know.
Maybe.
I don't know. Now, can we just go back a little bit before we wrap up and talk about the hype? And kind of what we know, maybe not for sure, but what we think we know about the microbiome, what we don't know, and where the hype's been justified and where it's probably not. So what's your take on all of this?
I think we're coming out of the hype now. So like I say, 20 years ago it was a niche area, and then it all exploded, and suddenly every disease could be cured with the microbiome, and it was everything. And people jumped to very quick conclusions. And I think now the field has matured. So that's pretty much, we're past that now, I think. But what is still a big problem, which is maybe somewhat related to it, is that people just looked at the changes in the microbiome, let's say in health compared to a certain disease, or the classic one is lean versus obese, and then they saw a change and they immediately jumped onto the conclusion, the bacteria make you fat or something like this. And that is still happening, and it's still, we haven't managed to get rid of this in the research field, that people haven't mechanistically shown the actual causal relationship and jump to these conclusions. It's got a bit better, but still papers published, and you see it in the title, whatever, blah, blah, disease can be cured by this bacterial change. And then you read the whole paper and you can't find any mechanistic link there. They literally have seen as a change in the microbiota. Yeah. And then they're concluding that this causing the disease. But of course, and obesity is a good example, because, what you eat will affect how much body fat you have, and it can also affect the bacteria. It doesn't necessarily mean a change in both. It's actually causal link between the two. So, and that is infuriating. It still happens a lot, but that also, I think, contributed to the hype, because for a while everybody said, bacteria changed, and didn't really see what other factors could have changed the bacteria side by side with the disease development.
You must think it is very important, because you've been working in the field for a long time. So
I do.
So what do you think then are the kind of tangible learnings about the microbiome in terms of disease and health that we've taken away? Like what's real and what's not sort of smoke and mirrors?
So I think from a healthy person perspective, if you eat very little dietary fiber, you will likely not be as well and healthy. Both. I think you will probably feel not so good day by day. And I think there's a lot of evidence that shows that very clearly. If you eat sort of junk diets all the time, which in Britain is now very, very high, isn't it? Is it over 50% of all food eaten is
ultra-processed?
Yes, and takeaways
crazy,
and stuff like this. It's crazy, you know.
It's really crazy.
So that definitely is bad news. So you do need to eat that diet to be really well. And then also you may set yourself up for getting cancer down the line or heart disease. So definitely you will preserve your healthiness and your well-being much more. And there may be some extremes. You talked about Magrani, who was smoking heavily and all this. Some people probably get away with a lot of bad diet lifestyle. The same as alcohol. Some people can drink all their life and never get that liver cirrhosis, and others hardly any. So it is very personalized. And again, the personalized thing obviously will come much more now in the future, that we understand all this much better. So it's the whole system we need to look at. Yes, if you want to stay healthy and well, eat a varied, mostly plant-based diet, I would say. I personally am not a vegetarian or vegan. Again, my philosophy is we weren't evolved. And I don't want to take supplements because I'm worried about not getting enough B12. So I eat meat maybe once a week or something like this. And I'm not fussed about whatever. If I'm invited or something, I eat pretty much what's been given to me. But mostly I live off plants and good plants and brown bread or, whole wheat bread and all that sort of stuff. But I'm not religious about any of these things because, again, for me personally, with my personality, I think it would stress me out so much to get too absorbed in having it absolutely perfect, because I think in the bigger scheme of things, it's not going to make a huge difference. If it shaves 5 days of my life because I've sometimes let my hair down or don't worry about it too much, I think that's fine, So that's my philosophy. If you have a disease, then the microbiome could have a huge effect. That's a whole different ballgame.
Now, as we kind of wrap up, are there any sort of key messages, learnings, statements that you think might be helpful for the listeners, in terms of the work that you're doing, fibre, prebiotics, probiotics, polyphenols? We've talked about the hype, and I think that's really helpful. We've talked about the diet, eat a variety of mainly plants. Is there anything else you think might be quite impactful?
I think we probably covered it all. But I would say, eat a healthy diet, a varied diet, mostly plants. Don't stress about it too much. But don't let your hair down every day either. I think if you are feeling well and you're eating well, there's no need for pre or probiotics. Also supplements, we touched on that earlier. I try to limit my supplements because I'm worried about potentially overdosing.
That's really interesting.
I don't know whether enough studies have always been made. At the same time, I do take supplements that I think I'm probably lacking in. Vitamin D, probably everybody should. I take the lowest dose and only vitamin D, not multi, this and that and the other. And again, for somebody like me who doesn't like fish very much, I think fish oils are good to help me with the old arthritis and all this. So there may be supplements that for you personally with little things you have work out well. But I'm not thinking that, sorry James,
it's fine.
That it's maybe the best thing for your health to pop the pills like hell because you
really
think more is always better. But I may be wrong, We'll see. We'll talk again.
We shall see. We'll talk. Well, I don't think you've
maybe we need to compare telomeres at some stage.
We could do that. That'd be interesting. I don't think you've actually aged at all since we first met. So
you're not looking very closely.
I think that's maybe a result of this plant-based diet. I, however, have aged because I saw.
You're looking very well. I think.
Well, thank you. It is actually my birthday today.
Yes, I forgot that. Happy birthday.
And I am aging because there's some grey coming in, but I'm working really hard on staying as fit and healthy as I can. And I do believe in the microbiome. Fundamentally.
Me too, absolutely. But as a part of the whole system, right? I'm not one of these religious microbiome is everything and nothing else matters. And I think from my personal experience, also looking around my family, genetics can be so important. And like I have in my one family, I have the two extremes,
Yeah. And that's something we can't change. We can change our epigenetics. We can change our microbiome, which interacts with our genome.
Well, we will be able to change our genome. And, that's also so exciting that all the fields are maturing. I mean, when a human genome came out And then there's this big and nothing happens for years, and now we're really
happening.
reaping the benefits. So I think on all aspects, we're going to see, if humanity is not messing everything up big time, we're going to see.
We are.
Yeah, I know. What I'm saying is, if we're here to see it with our
better,
yes, exactly.
There's good and not
so
good,
I think that's the nice thing. It's all coming together now. With this whole personalized nutrition, for example, we talked about it for a long time, but I think the building blocks are starting, and we get the tools and technologies and knowledge all coming together that maybe not in 5 years, but we will in the future know what is important for which person. For one person, the microbiome may be the one thing that's the main driver of them not being well. For another person, it's maybe a gene that's faulty, and we may have the tools to repair that, or we can give them effective medication to counteract that. So, it's one aspect of our body. Mm-hmm. I think it's as important maybe as your liver or something, but you can't say it's just the microbiome or everything else. In that sense, actually this analogy with the gut being another organ, as in the gut microbiota being another organ, is quite a nice one, actually. It's just more malleable than your liver, which is nice.
And that's very exciting.
Yes.
So on that note, we shall conclude. Wanted to express my gratitude and thanks, both for coming on the podcast, but also for all the help and support you've given me and the company over the last, how many years now? Long time. But as I'm sure you can see, we're working very hard. And, during the early days in particular, the support that you gave me, because you were actually the first person I spoke to, and laterally others, was fundamental and instrumental. It wouldn't have happened because I didn't have the credibility, and you all took a bit of a bet sitting on this advisory board of this crazy student. So thank you very much. And we're getting closer every day to an approved drug for patients.
Great.
That's super exciting.
It is. Well, thank you very much for inviting me. It was a pleasure.
Pleasure.
I have to say I enjoyed watching your journey from start to, where we are now. So,
thank you.
Very well done, and I'm very excited for how things are going.
Thank you, Petra. Much appreciated.